Okay, good morning. Today we're going to talk about some more chromosomes, starting with the amazing Y chromosome and then going on to some chromosome aberrations which are important for human health. Important parts of the Y chromosome. We're just going to deal with three parts specifically. First is the pseudoautosomal region, which pairs with the X chromosome. Then we're going to talk about the SRY region, which is the male determining region of the Y. And then we're going to talk about the ampliconic genes that are important for spermatogenesis. So taking it from the PAR region, the PAR refers to pseudoautosomal, and it's pseudoautosomal because it does in fact have a homologue on the X. Most of the genes on the X and Y are not the same, and so you wonder why we can treat them as a pair. Why would they pair in meiosis? Well, they do pair because of the pseudoautosomal region that's common to both of those chromosomes, and that allows the two chromosomes to act as a pair in meiosis. In fact, there's an obligate crossover that occurs within that region that keeps the two chromosomes together so that they will segregate as homologs properly during the meiotic divisions. In general, genes in the PAR region are considered pseudoautosomal because, for one thing, they largely escape X chromosome activation and therefore do exist on both the X and the Y. So they behave as pseudoautosomal genes in the sense that each gene that's there has a homologue on the other chromosome. Okay, so why this can be important, and people are still thinking about this, the Klinefelter males have XXY karyotype. They have three active copies of the PAR region, two from the X's and one from the Y. Similarly, or conversely, Turner females have only one active copy of these genes. So there are in fact dosage differences versus normal karyotypes in the functions of these genes. So we do wonder why sometimes there is any phenotype at all for Kleiner and Turnfelder when we do have X chromosome inactivation. And the differences in expression of these PAR region genes could actually be part of that mechanism, why there's a phenotype in these individuals. Okay, so here's a picture of the Y chromosome again, showing the pseudoautosomal region on one end of the chromosome. Now, immediately adjacent to that is the SRY region, which is the sex determination region of the Y chromosome. So this is an amazing gene, actually. It encodes a transcription factor, and its job is to activate more than 2,000 autosomal genes. So that is a very important transcription factor because it's involved in things like development of the testes and spermatogenesis. It's kind of amazing to think that females have all of those 2,000 autosomal genes and therefore are perfectly capable of developing testes and sperm, but because we lack this particular transcription factor, we do not do this during embryonic development. Those genes are not expressed because the SRY transcription factor is not present in females. So this is the sex determination region on the Y. So the third important part of the Y chromosome that we need to look at is the region indicated on the diagram where the so-called ampliconic genes are located. Okay, now the Y chromosome is obviously very important because you need to have those genes in order to actually complete spermatogenesis. But having no homologs on the X, that means that a single mutation in one of those genes could cause a person to not be able to form viable sperm. Well, what's happened here, that's illustrated on the diagram, is these ampliconic genes, is that each one of those has itself a backup copy in usually an inverted orientation relative to itself. So if you read along the bottom of the slide where the little arrows are pointing in opposite direction, the genes that are located in those regions are in fact repeated in inverted order relative to their own homologue in essence. So that we have a backup copy of each, and that actually has a way of rescuing, besides if you have a mutant, making it not that you're going to be unable to produce sperm, but that actually provides a genetic mechanism for fixing or recovering the function of those genes should they be mutated. I think I probably said all this. So the same protein coding regions are present in inverted orientation relative to each other, and these are incredibly crucial for spermatogenesis after the testis has already been formed during development. Again, look at the diagram. There are deletions of the Y that lead to infertility and low sperm count. Okay, one shows a region where men having fertility problems turn out to have a deletion on some copies of their Y chromosome. Not necessarily all. This would be something would have happened during the life of the individual, the life of the man, that some of his cells may, or some of his Y chromosome may be deleted for this particular region. There's another region called the GR deletion, which doesn't take out quite as many of the genes, so that leaves at least one copy of each. So this would lead to lower spermatogenesis, but wouldn't knock it out completely. Okay, so if your dad had inherited a B2, B4 deletion, you wouldn't be here. And so a lot of these occur, they're de novo mutations occurring during the life of the dad. We're okay. Okay, moving on, we're going to talk about translocations. Remember, there's different kinds of alterations that can occur in a chromosome, not necessarily in our germline. And we've talked a little bit about when you have extra chromosomes, gains and loss through chromosome non-disjunction, for example, during meiosis. So now we want to talk about translocations, where parts of the DNA has been moved to another chromosome, often reciprocally, where two non-homologous chromosomes have changed parts. Now, this doesn't sound like a terrible thing, and usually it is not, but it can cause disease, and it can certainly cause problems in gametogenesis and in creating progeny. Okay, so we already talked about trisomies and monosomies. Today, we're mostly going to talk about translocations. We're going to mention inversions and discuss something about how the segregation of translocated chromosomes in meiosis leads to imbalance and problems with the progeny. Okay, so we already saw chromosome translocation once, and this is just to remind you that the CML, chronic myelogenous leukemia, is caused in large part by a reciprocal translocation involving chromosomes 9 and 22. In this particular chromosome translocation, part of chromosome 9 containing the ABL proto-oncogene is swapped with and is broken with the chromosome 22 BCR region, breakpoint cluster region. So those two chromosomes, 9 and 22, have simply exchanged parts of their long arms in such a way that the BCR region and the ABL proto-oncogene are now in juxtaposition, leading to overexpression of the ABL1 gene, and this leads to the CML. So this is not an inherited translocation. It's something that happens during the person's life, but it does create problems because it activates this proto-oncogene. Okay, so in general, part of a chromosome is moved to another non-homologous chromosome, and these may occur during life of the individual, or they can be inherited. First of all, they are very important in evolution because they help isolate populations from each other, and there are very many interesting things having to do with translocations in evolution. Okay. All right, so we're going to look at the human and chimpanzee Giemsa bands in order to see what chromosome rearrangements are different between human and Giemsa. It's kind of amazing, really. Okay, so this slide shows the human chromosomes lined up with their chimp chromosomes, and we can see we've got human chromosome 1, chimp chromosome 2, 3, 4, etc., lined up next to each other. And the human and chimp Giemsa banding patterns are really very similar to one another. They're not quite identical, of course, but you can clearly say which human chromosome is similar to which chimp chromosome and line them up according to that. The one outlier here is chromosome 2, where human chromosome 2 is really much longer than the chimp one. The one lower arm, the Q arm of the chromosome, you can see the banding patterns are really very similar, but there's this missing part on the upper part where the human chromosome 2 is greatly elongated compared to the chimp one. Well, the answer here is the lower right-hand part of the picture where the remaining part of the chimp chromosome 2 is found. And it turns out if you take that banding pattern on that human chromosome 2 and chimp and compare them, they are actually very similar to one another. So it is believed that human chromosome 2 was formed by fusion of two chromosomes in our ancestors. So the genes match here, and we're short a chromosome relative to our chimpanzee and gorilla relatives because of this chromosome fusion, which occurred in the human lineage. Okay, so this is not quite accurate because it makes the bands look much more similar than they really are, but it makes the point that the human chimpanzee chromosome, there used to be two of them, was fused together to make one long chromosome, which is now human chromosome 2. So these are called chimp 2A and 2B because of their similarity to actual human chromosome 2. Okay, so it came from the fusion of two chromosomes in the lineage leading to humans, and it's a translocation because of regions of non-homologous chromosomes exchange parts. Right, so here's what this looks like in evolutionary terms. There was believed to be a common ancestor having 48 chromosomes, and the 48 chromosomes persists in the gorilla, bonobo, and chimpanzee lineage. But in the lineage that led to the humans, Neanderthals, and Denisovans, that chromosome fusion occurred and does distinguish us, our chromosome constitution, from our other primate ancestors. Okay, so translocations in general and their effects. If you move a big piece of DNA from one chromosome to another and you haven't gained or lost information, it really shouldn't matter, but sometimes it does. You've got the right number of genes in the right quantities, but it does affect meiosis depending on if you're a homozygote or a heterozygote for a translocation. And as we saw with the CML, the translocations in the wrong place, that can be very bad. So imbalance in the gametes leading to genetic disease in the progeny is what we need to discuss now. So the idea is you've got a person who's got a translocation, but nonetheless has a balanced genome. When their chromosomes pair and segregate, you can have problems in the progeny. Okay, so our first example is going to be a Robertsonian translocation, and these are kind of interesting because they actually involve the acrocentric regions of small chromosomes in which the extreme part beyond the centromere consists largely of repetitive DNA and not much coding information. So that part of those acrocentric chromosomes is actually dispensable in terms of the life of the human. So what has happened in a Robertsonian translocation is those two acrocentric regions have been disposed of by means of a translocation, leading to the fusion of the two chromosomes involved. So we've now got a yellow chromosome and a green one, and somehow they've become fused into one large chromosome, with very little loss of information and no gain of information. And that's okay, except that this type of translocation in human is fairly rare. Most people, of course, contain two normal copies of chromosome 14 and two normal copies of chromosome 21. Remember that Down syndrome arises from extra copies of chromosome 21 information. Now think about what happens if you have a person where their chromosome 14 and one of their chromosomes 14 and one of their chromosomes 21 fuse together to form one big chromosome. Now that person still has the right amount of genetic information. They've got two copies of everything on chromosome 14, and they've got two copies of everything on 21. So they've still got the right amount of information, even though the number of chromosomes has actually changed. This person has 45 chromosomes instead of 46 because those two chromosomes have fused into one. Right. So this can cause problems if somebody, when that individual undergoes meiosis, because the chromosomes can become confused as far as what segregates where, and that can lead to imbalancing in the gametes. Okay, so what I drew here is a picture of pairing of chromosomes 21 and 14 in a person who's, as it looks like, heterozygous for that translocation. Right, there's one translocated chromosome and there's one each of the normal chromosomes. Well, these guys are able to pair during meiosis because they are homologous. The 14 normal pairs with the 14 part of the long fused chromosome. The 21 pairs with the 21 part of the translocated chromosome. So far, so good, but problems arise when the gametes are formed, depending on how the chromosome 21 segregates. Now, I should point out that I'm representing a bivalent here, And I'm not showing all the chromatids. I'm just showing the chromosomes as a line. I'm not showing that each one actually has 2 chromatids on it. So you're going to have to forgive me for that one. All right, so if we segregate these chromosomes, there's 2 ways in which that can happen. One which gives normal progeny, and the other gives what's called translocation downs, because those children have an extra copy of 21 information. So that's shown better on the next slide. Okay, so this is a way that chromosomes will segregate to produce translocation down syndrome. Okay, what happens is the chromosome normal 14 and the normal 21, if they segregate together, are going to lead to balance. That's not a problem. What happens is if the translocated chromosome, which is the 14-21 fusion, segregates with the normal chromosome 21, that gamete ends up with extra chromosome 21 information. So I'm showing that here. We've got a 14-21 fusion plus a 21. So this gamete has 2 copies of 21 information, not just one. That's going to be a problem. The other product of that meiosis will have a chromosome 14 only, no 21 at all, and that's going to be a lethal situation. So to get the possible genotypes of the progeny, we'll have to consider that the other parent will contribute a normal gamete, and that will contain 1 normal 21 and 1 normal 14. So that's represented in the lower part of the slide. This individual got the 14-21 from the translocation parent, a 21 from the translocation parent due to segregation and meiosis, and they also got a 14 and a 21 from their normal parent. Now that person has 2 copies of 14 information, no problem there, but they have 3 copies of 21 information, and that leads to translocation Down syndrome. The other product of that meiosis gets a normal 14 from their normal parent, a normal 21 from their normal parent, but they only got 1 14 from their translocation parent. So that's a lethal situation because the individual is totally missing the copy of 21 that should have come from their other parent. So we're essentially monosomic for chromosome 21, and that is a lethal situation. Okay, reiterating, the balanced parent is a normal 14, normal 21, and the fusion. They pair, lining up as with their homologous sequences, and the problems occur when they segregate. That if you get the 21 going with the fusion chromosome, you end up with extra 21 information in that gamete, with the result that you end up with Down syndrome in the child. Of course, the other gamete has no 21 at all, so that offspring would be monosomic for 21, and that's an inviable situation. Okay, this just depicts Robertsonian translocations that actually do occur in human. One that does occur is, of course, this 21-14 fusion, and shows the karyotype of the carrier individual, 1 normal 14, 1 normal 21, 1 fusion. And the child that gets the trisomy 21 is the one that got where the 21 segregated with the fusion chromosome. Similar situation can occur with chromosomes 13 and 14, which are also acrocentric chromosomes. Those 2 have fused occasionally in the population, and that leads to, by the same mechanism, essentially trisomy 13 information in some children. Now, it's probably not fair to call this trisomy because there's not an extra chromosome necessarily, but it's equivalent to that in that you have extra information. Okay. Crete-Shaw syndrome is another example. It's not the result of Robertsonian translocation, but rather a reciprocal translocation involving chromosomes 5 and 15. Now, this can be inherited, but it can also result from new rearrangements in one of the parents. Okay, so the basis of this is that part of chromosome 5 has moved to chromosome 15, and that a very small part of 15 has moved to 5. So this creates a situation where you've got a translocation heterozygote, and you end up with disease depending on, again, segregation of those chromosomes during meiosis. Okay, so to simplify this, we're going to say that 1 copy of chromosome 5 has lost a little piece. That's the part that moved to chromosome 15. So we're going to call that 5p minus, meaning that that chromosome 5 is missing some information from its p arm. Okay, the information from chromosome 5 is still present. It's moved to chromosome 15, so we're going to call that one chromosome 15q+. So we've got the right amount of information. It's just that it's rearranged compared to a normal karyotype, but it's not going to cause any problem in the person. It'll cause problems in the progeny, as we'll see. Okay, so what I drew here is pairing between those two aberrant chromosomes. Just as before, chromosomes are pretty good at finding their homologs, pairing with them in meiosis. And in this case, we have the normal 15, the normal 5, pairing with their translocated counterparts, where the 15 part lines up with the 15 part, but it's got a little bit of extra information on its end, and that's the part that matches chromosome 5. So chromosome 5 matches up with that, and meanwhile, the chromosome that's deficient, the deficient chromosome 5p minus, lines up with its homologous sequences on the normal 5. So everybody's paired and everybody's fine. And again, there's extra chromatids I didn't draw in. Everybody's paired, but the problem is going to occur depending on how these chromosomes segregate. The parent was balanced, but some of the children will be imbalanced. Right, so this is just what I just said. You get normal pairing, but what happens is what causes the issues is when some of the time you get segregation leading to imbalanced gametes. Okay, so here's the pairing arrangement, and I'm just going to illustrate when you get a normal situation, right? It's not always a problem. You can pair everybody, segregate the normal 5 and 15 together, segregate the Q plus and the P minus together. So the progeny from this gamete will be balanced, just like the parent. They lost information from 5, but it's present on 15. So altogether, it's a balanced situation. So when that happens, you have fertilization with a normal parent, and then you can have, depending, the normal 5-15 segregation gives you normal 5-5-15-15. But the one where you got the Q plus going with the P minus, that gives you a individual, which is just like the parent. They have the translocation, but they're normal because it's balanced. Okay, the other outcome of meiosis, the one that produces disease, is where the P minus chromosome segregates with the 15q plus. Okay, in this particular case, we line up our chromosomes as before. Homologous sequences can find themselves in pair during meiosis, and then chromosome segregation occurs. Okay, in this instance, we will have the chromosome 15 going along with not the 5 itself, but with the 5p minus chromosome. Okay, so when that happens, we have a 5p minus with the 15. So that gamete is actually missing that end part of chromosome 5 that was chopped off and moved to 15q plus. So we're missing part of 5 in this gamete. The other half of this segregation pattern is when you have 15q plus going with the normal 5. In this case, we have the 5 information is present on the normal 5, but it's reiterated on the long arm of chromosome 15. The plus also contains 5 information. So instead of having just one copy of all of 5 information, that little part that's stuck on 15 is there twice in this particular gamete. So when fertilization happens with a normal gamete, the first pattern gives Crete-Shaw syndrome. In that one, we have the 5p minus and the 15 together, fertilized by a normal gamete, gives us 5p minus, 15, 15. Okay, so that person has only one copy of the chromosome 5 information that's missing from the short arm, the 5p minus. And that's the one that gives the Crete-Shaw syndrome. It has only one copy of the genes on the extreme end of chromosome 5. The other one, which is the resulting from the 15q segregation, that individual has a normal 5 and a 15q. So that gamete has more than one copy of that. There's 2 copies of that extra information on 5. It's not the whole 5, it's just the part that's stuck on the end of Q, of 15q. But when that's fertilized by a normal, we have 2 normal 5s, a 15 and a 15q plus. Now that q plus represents extra 5 information, so that individual has 3 copies of that end of chromosome 5 that was stuck onto the Q plus. So that actually turns out that that's lethal, not, as maybe you'd expect, partial trisomy for this small part of chromosome 5. Those children are not born, and that's a lethal event. Okay, so summary of this. 5p segregates with 15, and you're going to get a Crete-Shot syndrome child. And the 5 with the 15q plus is an inviable situation. Okay, here's a karyotype, a picture of what those chromosomes look like. Here's a Crete-Shot normal 5 paired with the Crete-Shot, the 5p minus, and the deleted region is shown in the ideogram. Here's a full karyotype showing the same thing, where we have a normal 5, and a good cytogeneticist could tell easily that other chromosome 5 is missing part of the P arm. Okay, which of the following results from imbalance due to translocation? Turner syndrome, Crete-Shot syndrome, triplo X syndrome, trisomy Down syndrome. Yes, the answer is B. Okay, I'm going to just say a few words about inversions and their consequences, just so that you know what they are and that they can cause problems. They're actually very important mechanisms in evolution as well, and we're not going to have time to go through the consequences, but I'll just point out what it looks like. So what's an inversion? An original chromosome, let's say, is A, B, C, D, E, F, but a chromosome that's had an inversion might read A, D, C, B, E, F, where B, C, D has been flipped over and reattached, and so now it reads in the opposite direction. So this actually messes up chromosome pairing because the genes are not in the right order, and actually the chromosomes can amazingly twist around each other to let pairing occur correctly. So that's not the part that goes bad. The chromosomes are able to contort themselves using this thing called an inversion loop, where the B, C, and D part of the inversion can actually pair with their homologous sequences on the other chromosome. Notice all 4 chromatids are present in this particular picture. Okay, the crossover causes problems, however, in that when you do have a crossover within that fairly large region, which is a very likely situation, the results can be devastating. You can actually end up with broken chromosomes and losing bits and pieces of chromosomes, etc. So crossing over with an inversion does cause inviability in at least some of the gametes. So it's way bad, causes loss of viable gametes, reduces the production of offspring.